Hello again, and welcome back. I'm now going to take a look at long-term potentiation, the basis of learning and memory. So we just heard about HM, his major contribution being showing where to look in the brain for learning and memory. Scientists then went to animal models to get at the molecular and cellular mechanisms. Now this immediately raises a problem here. Supposedly you're the scientist. How would you establish that the rat hippocampus is involved in memory processes? Because it could be that that was something perhaps unique to the human. So if you give this a little thought, I think that most of you will come up with the idea, well, remove the rat hippocampus, a hippocampectomy, and see if the animal has memory problems. So that's pretty straightforward. Not so straightforward, perhaps, is the next question. How do you determine if the rat has a memory problem? Now, there was many creative solutions over the years have come up for this problem, but one of the earliest and what is considered often one of the best is something called the Morris water maze, named after Professor Morris, who first used it and developed this system. And what you have is a rat or a mouse swimming in a tank of liquid. The liquid is opaque. Often they use milk for this purpose. What you don't see there in the picture is that there is a hidden platform. I'm just illustrating it down here. So here is our rat or mouse swimming around, and just below the surface of that liquid is a platform. Now the rat or mouse, it doesn't like swimming around, and it's madly looking for that platform. Now the trick here in the Morris water maze is to see if the animal can remember where the platform is. And so here is a typical kinds of results that you get with the Morris water maze test. Latency. This is the amount of time it takes for the animal to find the hidden platform. Now, in this particular animal, it's taking something around a minute the first time to find that platform. It has no idea where it is. But on the next trial, you'll find that, hey, it's doing it much quicker. And improvement with the next trial, etc. So the animal is really learning and remembering where that platform is. So this is a pretty smart rat here. These are the kinds of results that one might expect for a rat that's not so smart. And in fact, in the early days of this kind of research, hippocampectomies were performed, and these were the sort of the results you would get with a hippocampectomized rat, whereas the normal solving the problem much faster. So it was indeed the case that the hippocampus in the rat and the mouse is required for learning and memory. What I now want to do is actually show you a video of the Morris water maze in action. This is going to be the rat on its second trial and on trial 18. And I'd like to acknowledge that these videos are courtesy of Ineke van der Zee from the Department of Cell Biology in Nijmegen. So click here to get this first video going. So here is our rat. And you can just see under there, I don't know if you have the resolution to see it, platform is right there. And there he goes, swimming around. This is going to take quite a while. The rat here is having trouble remembering exactly where that platform was. Just a little patience here, and he'll eventually get there. No, not this time. Yeah, he's getting close, but still having trouble. There he goes. Okay, now we're going to go over to trial 18. Here he comes. Well, he has really learned where that platform is. Okay, so again then we went from HM, we went into the animal models, and with this simple Morris water maze, it could be shown that the hippocampus in these animals is required for learning and memory. But now we want to get at the molecular and cellular mechanisms. What's going on in the hippocampus during the construction of a memory? And for that, we have to go in vitro. In vitro, it means in glass. In other words, what is done is that the hippocampus is removed from, or the brain is removed, and then the hippocampus is removed from the animal, and we look in vitro at what's going on in the neurons in the hippocampus. So to do this, just to illustrate to you the kind of dissections that have to be done, here is the whole brain of a mouse. That's the OB, that's the olfactory bulb, and here is the neocortex overlying it. And what is done is the neocortex is peeled back, and because deep underneath the neocortex one finds the hippocampus. And so we're seeing the hippocampus coming out now. There it is there. And what is then done is it's cut out with very fine scissors. And so we now have this tiny little hippocampus dissected out of the brain. And now we make what's called hippocampal slices of this tissue. And basically what it is, you can think here, if this is the hippocampus, you just slice it up like you would slice up a salami. So you make very, very thin slices of the tissue. And these tissue slices, this is one here, they are then brought in vitro. So this is in medium that has all the nutrients to keep the tissue alive. And now what we can do is conduct some electrophysiology on this tissue. We can look at the electrical activity of neurons in this tissue to see if we can find any sort of activity that might have something to do with learning and memory. So you can bring in stimulating electrodes to stimulate neurons. You can bring in recording electrodes to record the electrical activity of the neurons. And this is all done. This is a tiny Petri plate with the tissue in it, and this is brought under a microscope so that you can bring these recording electrodes directly on to the cell bodies of the neurons in the tissue. And with these recording electrodes, you can then record the excitatory postsynaptic potentials of the neuron, or the EPSPs. You can also record the action potentials. Now I'll be describing this a little bit later, but basically what you can do with this setup, and what was done with this setup, was this LTP was demonstrated, the long-term potentiation, which, as I've mentioned, is the core mechanism of learning and memory. So what I now want to do is actually demonstrate how LTP was demonstrated. So this is now going to be our demonstration of long-term potentiation in a hippocampal slice. The neurons in the hippocampus, they're called pyramidal neurons. This is an example of a pyramidal neuron. This is the dendrites here. This is the axon. Here we have the cell body. And what is done is with that microscope, and a recording electrode is brought down onto this neuron so that you can measure the excitatory postsynaptic potentials. Now, these neurons in the hippocampus, they receive innervation and synapses from other neurons also within the hippocampus. And the axons that are coming and synapsing, these are called the Schaefer collaterals. They come in, and I'm just showing one example here. All of these neurons would have Schaefer, all these synapses here would have Schaefer collateral synapsing with it. But here's one example. And what is then done is a stimulating electrode is brought into the tissue. The idea being, you stimulate here action potentials, you'll have release of neurotransmitter and activation of this neuron here to produce an excitatory postsynaptic potential here in the cell body. So here is a stimulating profile, and here is our recording profile. And here we then give a depolarization. Here's our depolarization to stimulate the Schaefer collateral. What we have here is nothing. Now that's a big disappointment. What the problem here is, a typical pyramidal neuron is receiving some 10,000 to 20,000 synapses. What I'm showing here is the cell body here and the dendrites of a pyramidal neuron. These little tiny little green dots represent the synapses, these 10,000 to 30,000 synapses. And in this case, we were just very unlucky that the stimulating electrode did not hit a Schaefer collateral that is innervating this neuron. So, unlucky. But what you simply have to do then is move your stimulating electrode a little bit, try it again. Now this time, what we see with this stimulation, we get an action potential, we will have release of neurotransmitters here in the synapse. This will activate ion channels, receptors on the postsynaptic side, and will lead to an excitatory postsynaptic potential. And there it is, the EPSP. Okay, so having had that result, we now know that this Schaefer collateral is in communication with this neuron, and we can actually measure the strength of the excitatory postsynaptic potentials. So what we do now is, about every minute we give a stimulation, and about every minute the consequence of this stimulation is an excitatory postsynaptic potential. What is done to demonstrate long-term potentiation is that a graph is made. You plot the strength of the excitatory postsynaptic potential against time. So here is our first 5, and the strength, sometimes it's the slope, or it's the area or the height of the excitatory postsynaptic potential. And there is our first 5 there. And we just keep carrying on. We go for a full hour, and you can see here that the strength of these excitatory postsynaptic potentials remains rather constant. And now, sometime around 1 hour into the experiment, we give a stimulation profile where we give a high frequency discharge. This is just a stimulation in very high frequency. This is called a tetanus. And what we will see here on the recording is we see here our excitatory postsynaptic potentials. This is where we have given the tetanus. Everything just goes off scale here. And then here's the EPSPs after the tetanus. So if we plot these two now, I'm going to show you here these two. That was the two just before the tetanus. And now here comes the tetanus, and now we're going to plot these two after the tetanus, and what you'll see is, hey, they're much stronger. Okay, following that, we continue with giving a pulse, a stimulatory pulse, about 1 every minute. And what we see here is our excitatory postsynaptic potentials in response. So here's the next 5 I'm going to plot here. There they are. And now you just can carry on for hours or even days. And lo and behold, this stronger excitatory postsynaptic potential, it remains strong. And this is where the term comes from then, long-term. We have a long-term potentiation. It's been potentiated, it's been made stronger. So that is then LTP. Now, question. What is happening here in this synapse to generate this stronger response? Well, that is going to be the subject of our next video. I'm going to explain how this LTP is generated, and that is in fact through the work of the NMDA receptor. So you'll see that in the next video.